So, good afternoon or good morning or good evening, everyone. Welcome to Brain Matters webinar series edition 13, Computing the Brain. This Brain Matters has been launched by the Human Brain Project and in its third and final phase. And it's a webinar series which explores the various issues being tackled by the Human Brain Project scientific community. The hour-long sessions focus on different areas of brain research and feature expert speakers with the goal of highlighting the HBP's scientific achievements and the state-of-the-art services offered by its infrastructure for brain research, EBRAINS. So my name is Rebecca Wade. I'm a professor at the Center for Molecular Biology at Heidelberg University, and I lead the Molecular and Cellular Modeling Group at Heidelberg Institute for Theoretical Studies. And I'm going to be moderating this episode of Brain Matters, Computing the Brain. So we are going to hear 3 talks, which have, in a somewhat different form, been presented at BrainComp 2022. So BrainComp is in its 5th edition, was started in 2013 and is organized jointly by the Human Brain Project, EBRAINS, University of Calabria in Italy, the Heinrich Heine University in Düsseldorf, and the Forschungszentrum Jülich in Germany. And it features international experts in a wide variety of areas related to brain research and high-performance computing, and is really aimed at discussing the computational challenges of brain connectivity and new approaches that are emerging from these studies. So, let's see if my slides were forward. So now, today we will have 3 speakers. Jean-François Mangin, Giulia Rossetti, and Sebastian Billaudelle, and they will focus on really quite a range of topics. So Jean-François Mangin will present new methods and approaches to investigating the principles of brain organization. Giulia Rossetti will talk about machine learning-driven translational medicine for the development of personalized brain drugs. And Sebastian Billaudelle will provide an overview of experiments and use cases for brain scales chip, which is an example of neuromorphic computing and uses brain-inspired technology for increased performance and energy efficiency. So each speaker will have 10 or 15-minute presentations, and then after that we will have a Q&A session. And we would like participants to write questions in the Q&A section, which we will then answer towards the end of the webinar. And please mention the name of the speaker to whom your question is addressed when you put it in the Q&A. And also please note that the chat will not be read, only the Q&A section. So before we come to the speakers, I would like to give a little bit of an introduction. And let's find my next slide. Okay, so the Human Brain Project is aimed at building a cutting-edge research infrastructure to support neuroscience. And this is sorry, I have a slight technical problem here. Let's just close that. And so, okay, so this is really a hugely challenging problem because of the huge scales covered when we think about the brain and what is covered in this project, ranging from the molecules that involve the proteins and others, right through all of these different scales here. And obviously there's a huge challenge in terms of collecting data, analyzing it and sharing it, but also in modeling it and then trying to understand it through simulations. And the project involves various different aspects, and you'll hear about 3 of these sort of cogs during this webinar. Now, just to give you some idea of how we can tackle these different scales, I'm going to concentrate on the area where my group has been working, related to subcellular and molecular simulations, so right at the bottom of the scales I've shown you. And even at this very small scale, we still have a huge multi-scale problem to deal with from a computational perspective. You know, suppose we want to understand brain plasticity and learning. This is really dependent on molecules that convey signals by binding to their various receptor molecules and initiating lots of signaling events. And we can ask, how can we simulate this? And what we try to do in the Human Brain Project is to use information about the 3-dimensional structures of proteins and other molecules and to simulate those in order to provide information that feeds into more abstract models, mathematical models of signaling cascades, provides parameters and information in order to construct appropriate topologies. And the idea is that if we can do this and generate models here, these mathematical models that allow us to understand important events related, for example, to learning or to brain diseases, then we can go back to the molecular level and evaluate what happens when, for example, drugs are applied or when there are mutations of critical proteins. And so this can, in the end, be used to guide diagnosis and therapeutics. So to give an example here, I show schematically a project that we worked on with 4 different groups in 3 countries in the Human Brain Project, where we wanted to look at key features for brain plasticity and model the particular pathway here with mathematical model. But this modeling was guided by simulations at the molecular level, even going down to quantum mechanical simulations of catalytic events going on in these molecules. And so we needed to combine a number of different computational techniques in order to be able to address this problem. And for example, in my group, we used a Brownian dynamic simulation technique to compute protein-protein association rates. And this involved use of the SDA software that we've developed over a number of years, simulation diffusional association, which allows you to look at the simulation, the diffusional motion of many proteins. But when computing parameters from such structures, it's really very computationally challenging because the timescales for these binding and unbinding events, when you consider the binding of a couple of molecules here, is on timescales that are much longer than the timescales you can typically simulate, even on high-performance computers with atomic detail models. And we also need to account for all of these motions, particularly in considering unbinding or off rates, which determine residence times of molecules. And so we need to have to also tricks to scale in time. And this illustrates the application of one approach we've developed in the group called random acceleration molecular dynamics, which allows us to look at how these drug molecules here unbind from a protein. You'll see that they go via different routes, which allows us to estimate residence times and to provide information that can be used to optimize the kinetic properties of such molecules. This is all described in more detail in this KB box, a toolbox for binding kinetics calculations. So one can also find information about how to do such calculations on the EBRAINS live papers, where we put together data for various of these different studies, such as the one I showed you a couple of slides ago. And we're also developing various databases and tools that allow one to access simulation trajectories or to generate them. And this is all gradually being put on the EBRAINS service infrastructure. So that by way of a short introduction, just looking at the sort of lowest level, the smallest scale that we are dealing with in the Human Brain Project. So now it's my pleasure to introduce our first speaker, Jean-François Mangin. He is the director of the Baobab research unit in Neurospin in the COR in Paris. And he's interested in neuroimaging data. And he's also a co-leader of the strategic human brain data project in the Human Brain Project, and he does that with Katrin Amert. And he's a director of the CATI, the Center for Acquisition and Image Processing, which is a French platform for multi-center clinical neuroimaging research studies. So Jean-François Mangin is going to be talking on the topic of what cortical folding patterns could tell us about individual brains. So now I'm going to stop sharing and hand over the screen to Jean-François.
Hello, everybody. Sorry. Here we are. Oh, no. Okay. So the pictures you see on the left hint at a kind of graal of computer chip design, namely packing memories and transistors in 3D. And this could speed up transit time much beyond what is possible with 2D design. And what you can look at the right, in fact, is the trick that nature has found to address this goal, namely folding. And the point today is that cortical folding is specific to each brain, and it's the reason why I will describe a research program that aims at investigating to which extent we can decipher this variability. Basically, we want to discover whether different architectures lead to different folding. So each of us has a unique cortical folding pattern, as unique as your fingerprint. Basically, I could identify your brain in a large data set of brains, thanks to this unicity. But if we look locally at the folding, in fact, we discover patterns that are a bit like letters or ideograms. And a bit like handwritten manuscript, each letter is written differently in different brains. You can find some interruptions, you can find additional strikes, and in some places you even discover that we don't all have the same letters. So what does it mean? And if you compare different primate species, you discover a kind of evolution of these letters. So basically, we are looking for a kind of hidden language that is explaining this variability and maybe meaning something about our brains. And this is not a mainstream research program. Basically, most of the people just think that this trick is just there to increase the surface area of our cortex while keeping the size of our skull, the volume of our skull reasonable so that we do not get into trouble during birth. But the reason why we believe that there's a strong link between folding patterns and architectures is because folding patterns occur in utero during the last trimester of pregnancies, just at the same time as neurogenesis and the differentiation of the cortex into architectonic areas. So there must be a link there. And at birth, in fact, the folding patterns are there. They will not change during the whole life, but the size of the fold is still changing. For instance, during childhood, the folds get deeper and longer. And as soon as adolescence, in fact, the process is reversed because of the development of myelin neuron axons. In fact, the cortex is unfolding. The surface, the length, and especially the depth of the fold is decreasing. And this is proceeding further until old age, either because we are losing some neurons to get wiser or because we are just following a pathological tracks, for instance, Alzheimer's disease. And all this change of the depth of the folds can be used as biomarker, but my topic today is the pattern that occur during pregnancy and will never change and give a signature of some specificities of our brains that occur because of development. And I would like to stress the point that I'm not claiming that folding patterns have some kind of function. It's not at all like the folding patterns of protein, because for proteins, in fact, it's the folding pattern that is creating functions, basically like if you have a key and you want to open a lock, it's the pattern of the key that is opening the lock. For folding pattern of the cortex, it's just a packing of all the neurons together that is leading to these patterns, but it's not leading to any function. And the interesting point is that there's a kind of planet alignment that is leading to a much more understanding of the origin of this folding process during the last decade. First, physicists have discovered that with a very simple mechanical equation, they can generate folding patterns that are very, very similar to what occur in real brains. What they model is the fact that the outside of the brain, mainly the cortical mantle, is growing faster than the white matter, the inside part of the brain, which is creating some tensions that are resolved by the folding. And they even push the equations further, triggering it from the shape of a baby brain, a premature baby brain without fold, and leading to this evolution with the simulation, looking even more like a real brain. And they even push the stories to the real world using two jelly with different properties. They created a brain with two layers, and they could activate the outside layer to make it grow faster than the inside layer. And just on the table, you see plop, 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 some folds appearing like a real brain. It's looking like a real brain, but the point is that the folds are not at the right place. And in each mammal species, there's a kind of stereotype map of where the folds should occur, and it's not with what is happening with the physicist model. Fortunately, there's another source of information, which is biology. And the biologists have discovered once again during the last decade what is making the specificities of folded cortex. In mammals, most of them have a folded cortex, but some do not have. For instance, mouse's cortex is not folded. And the difference is that between the ventricular zone where neurons are created and the cortical mantle, there's an intermediate zone where in folded mammal species, there's much more proliferation of neurons than, for instance, in mouses. And in fact, this process is not homogeneous. If you look at these sections of a ferret brain, you see that there are some places where you see this proliferation and it's not occurring in between. And basically, the places where you have proliferation of neurons will become convolution. And there's a kind of mosaic there that is leading to the folding pattern of the cortex, which is a bit like a protomap, a bit like the protomap that is supposed to give birth to the differentiation of architectonic areas. And the question is whether there's differences in this protomap leading to different folding patterns. So now I will explain you what we can do with computers to address this point. Here, it's a kind of a special kind of striptease. It's my brain. And when you look at a brain like that, you realize that first, that there's a kind of folding wavelengths, all the convolution have the same width, which is meaning that big brains are more folded than small brains, which is very disturbing. In fact, in big brains, you have more of these small folds in between, in the middle of convolutions. And if you remove the cortical mantle, you can have a look at what is behind, what is hidden, in fact, which is sometimes buried jarry in the middle of the fold that explain, in my opinion, a lot about the variability across brains. And you can ask the computer to compute a kind of negative cast of the brain. It's something a bit similar to this kind of surfaces you see in walnut here, surfaces that follow, in fact, the shape of the fold that can be manipulated by the computer, split into pieces in order, in fact, to recognize, in fact, the main sulci that have been described by anatomists for decades. In fact, there's about 100 of them. And now we have some deep learning systems that can give these colors to this negative cast according to the nomenclature of medicine. And once you can do that for thousands of brains automatically, you can measure some features of these folds, for instance, the length of the sulcus, the depth of the sulcus, or for instance, the distance between the two walls that make up the sulcus, and try to use it as biomarker. And it's working, in fact. For instance, if you take 10,000 people, for instance, from the UK Biobank dataset, and a specific sulcus, for instance, this red one here, which is called superior temporal sulcus, you can see that the distance between the two walls, the kind of width of the sulcus, is increasing with age between 50 and 70 years old. At the same time as the thickness of the cortical mantle surrounding the fold is decreasing. And if you look more carefully at the percentage of the population where this increasing of width is the fastest, you observe a kind of change of slope of this process, which is probably corresponding to something occurring in their brains that could be related to some pathological process. And basically, if you if you use, in fact, the opening of different sulci, you can use it to predict, for instance, conversion to dementia in old people. But my point today is not this variability occurring during a life. It's mainly the folding pattern occurring during pregnancy. And what I've been hiding up to now is this terrible variability, because I've been showing only one brain, my brain. But if you look at two brains, and you imagine yourself trying to create a kind of roadmap explaining the variability across these two brains, it's very difficult. I can help you adding in there what is called the central sulcus, one of the most stable fold, which is in fact corresponding to primary sensory motor areas. You see that it's very similar across the two brains. But if I look at another sulcus, for instance, here the superior temporal sulcus, you discover a lot of variability. For instance, you discover that in the front of the left brain sulcus, there's a kind of interruption that looks like corresponding to this stuff you see here, that are some other convolutions that are in the two sides of the sulcus, but did not merge, in fact. And you see the same process here. You have an interruption that is looking like connected to what you see on both parts of the sulcus here, but it's not the same organization. And in the back, you see once again that the back of the red fold is connected to the blue one here, while it's connected to the green one here. What is triggering this variability? What could it mean? And if you go to the frontal lobe, it's even worse. It's a kind of nightmare. It's very difficult to put this color, this nomenclature there. And there's a lot of disagreement even across experts, because the variability is just beyond reach for our cognition. So the idea is that during development in utero, the folding dynamics it can be attracted by different configurations. There's different attractors. And for instance, these two configurations of the superior temporal sulcus could correspond to two different attractors of these dynamics, corresponding to two different architectures or two different chronology of the maturation of this architecture. And another way to think about that is to use another way to simulate, in fact, the cortical folding process using what is called Turing equations. Maybe you heard about that. It's a very simple of partial differential equations that can be used to simulate various phenomena in nature. For instance, shells or different kind of skin patterns. And the interest of this equation is that they are built only upon two parameters. So you can do a phase diagram of the different kind of folding occurring according to the parameters. And you have a domain that is maybe what I could call ecological domain, leading to surfaces folding like a real cortex. Domains where you get some kind of spots, which is corresponding to what is found in nature too, in some very bad diseases, which is called polymicrogyri. And you can find domains where the cortex is not folding, and it's also something that occurs in nature in even worse pathology. Because of very thick cortex, the cortex cannot be folded. And what is interesting is that to think about the fact that in the ecological domains, you could have sub-attractors corresponding to these different patterns that I was mentioning before. For instance, interruption of the sulcus in the front or in the back could correspond to different attractors. And in between these attractors that are normal and the very pathological ones, there may be some smaller ones corresponding to events occurring during the development, leading to some weaknesses, or maybe some diseases like psychiatric syndromes. So another way to think about that is to use a metaphor, a link between the folding patterns of the cortex and text. Basically, texts are made up of words, and the words are made up of letters. And you could imagine that in the folding patterns of the cortex, there are some entities that are atoms that cannot be split, that we could call the sulcal roots, and we could associate each root to a letter, and the phenomena I was describing before can be understood another way. For instance, in this brain, where you have a very long red fold, which is the superior temporal sulcus, all the sulcal roots of the split of the sulcus did connect, leading to this long sulcus and a long word. And in this other configuration, there's an interruption here in the front, which means that a letter has been left alone, and you have another word which is corresponding to this configuration here, and so on for other patterns. And of course, because the brain is a very, very complex folded entity, there's a lot of these sulcal roots. There's a combinatorial explosion that is very difficult to master. But in fact, not all the configurations have a high probability. For instance, here, what you see is an average of 150 brains, which is called the ICBM template that is used a lot in the field of neuroimaging, which is corresponding to the most standard configuration with very long circuit without interruption. And basically the distribution of the probability of each of the configuration has a kind of funnel shape, which means that a few configurations have a much higher probability. And what we've been trying to do for years now is to do a dictionary of all the folding patterns that you can find in the general population, and to list the ones that have a high probability that can be considered as normal, so that we have a chance to define what is abnormal as patterns that are not present in the dictionary of the very frequent pattern that we found in the general population. And we do that with 50,000 brains these days. And for a long time, we've been very naive. We've been working on this topic with anatomists and developing incredible system for visualization and browsing all these data sets so that they can discover things that have never been found before. And it's basically very, very difficult. Fortunately, from time to time, we do a discovery. For instance, this one is interesting because it's corresponding to what's happening in patients who have epilepsy triggered by the sensory motor areas. And in a lot of these patients, my colleagues have discovered that there's a pattern that is ironically looking like the power button of computer, and very rare in the general population. But we have very few of this kind of it, just because this is so complex that it's beyond reach for our cognition, and it's where machine learning has a role to play. And I will show you a few examples of what we can do these days. For instance, I will be using the
Jean-François.
Yeah.
Can you draw to a close? We're getting towards the end of your time.
Okay, I will go to a close. So I will switch this result, and I will conclude by the fact that these days, in fact, we are trying to use unsupervised artificial intelligence to try to do the mapping of this dictionary, because the standard models are limited, a bit like the models of the hand cannot account for all the variability that you can find in nature because of genetic disease. So basically, what we are trying to do with deep learning is very similar to what has been done to understand the variability of the folding of proteins. We have a few 100,000 proteins where the folding is known, and this has been taught to deep learning to be able to predict what can be done, the way any protein is folding. And in fact, basically, we can do the same with the brain data from clinical research, where we have annotation, and we can use them as a kind of Rosetta Stones to link, in fact, the folding patterns with all kinds of pathologies to build up this dictionary I was hinting at. Thank you.
Okay, thank you very much, Jean-François. Our next speaker is Giulia Rossetti. And Giulia, I think you can share your slides. Good. Okay, so Giulia Rossetti is a professor for computational molecular medicine at the Department of Neurology in the Faculty of Medicine of the RWTH Aachen University in Germany. And she's also a group leader at the Forschungszentrum Jülich, at the Institute for Advanced Simulation and the Institute of Neuroscience and Medicine. And she's also affiliated with the Jülich Supercomputing Center. And so she is a leader of the molecular task in the Human Brain Project, and works on the combination of molecular simulation and data science approaches towards drug discovery, in particular CNS drug discovery. And so this is the topic she's going to be addressing now in her presentation. So, Giulia, the floor is yours.
Okay, thanks a lot, Rebecca, for your introduction, and good afternoon to everybody. So let's go to a different scale with respect to what we have seen in the previous talk, because what my group is mostly concerned with is molecular medicine. So we are at a molecular level. Of course, you can look at the brain and different kind of level. as well as disease can be seen at the level of the patient, at a system level, connectivity, brain cell. We look at diseases, and in particular at brain diseases, from a molecular point of view. And the reason why we are doing this is because even if drugs are prescribed and taken at organism level, they exert their function by interacting with their target at molecular level. And we do so by using an umbrella of technique by which we try to infer also what is happening at the different scales. So we use a physics-based approach. We use bioinformatics, chemoinformatics, artificial intelligence, and system biology. Of course, all this connected with experiment. Now, what we are trying actually to understand with all this approach is, are the molecular aspects of neurotransmission. In other words, we know that the synaptic transmission is a mechanism of neuronal communication, but it is actually the sum of several interaction events that happens at molecular level. When we have a neurotransmitter binding to a receptor, we have an entire chain of events that is happening at molecular level that determine the outcome then at the subcellular and cellular and then neural network level. So what we are showing, I will show in this talk is how we tried and we managed to connect the molecular level of this process to the subcellular one, and how this can be connected with the design of new drugs, as well as novel diagnostic tools. In other words, how we can go from molecular mechanism to precision therapeutics. To do so, we have chosen as a use case or particular category of neuronal transmitter that are the G protein coupled receptor. They are really, let's say, key node of cellular communication. Indeed, they are the targets of more than 1/3 of current FDA-approved drugs, and they are connected to several brain pathologies. Now, the GPCR-based drug discovery, structure-based drug discovery, have usually treated this receptor, unfortunately, like as black boxes. And the main focus of pharmacology on this receptor was based on finding molecules similar to the endogenous neurotransmitters. But we know now that there is more, and we know that there are several aspects that have been overlooked, especially from fields, from the structural level field. And the reason is that when we think about the event of a neuroactive ligand binding to a receptor protein, what we see at molecular level is only a single readout. That is, if the neuroactive ligand binds or does not bind to the receptor protein. But this does not tell us anything of what is happening after this neuroactive ligand binds, and it does not tell us anything about how this will be translated in cellular pathways and then therefore in the neuronal response. So what we are trying to do is to see if starting from molecular understanding of the ligand receptor binding, we can infer something at least the subcellular level. So how can we escape this readout that is only confined to receptor, to the binding event of the neuroactive ligand to the receptor protein? So what the first things that we did is try to understand, of course, how this is related with the subcellular pathway. So the first step is try to understand if we have a method from, to predict only with computational approach if a ligand is going to be an activator or in other words, is going to activate the G protein coupled receptor, or is going to inactivate the G protein coupled receptor. In other words, the first things that we did is try to understand if we can distinguish between agonist and antagonist of G protein coupled receptor only based on the structural level information. And we managed to do so by taking as a use case the adenosine receptor, that is one of the most well-studied GPCR, and we build up, let's say, or repository of all the known agonists and all the known antagonists, and we analyze the way in which they bind to the receptor, extracting or protein ligand fingerprint for each of the complexes. Namely, we build up all the possible complexes, and we embed this information in a bitwise encode interaction partner that has this kind of, let's say, vector 0101, and this have been used to build up a machine learning model, in particular a very easy model like a random forest model that was able to distinguish pretty, let's say, in a very good way, agonist type of binding with respect to antagonist like of binding. So we have a model now that can distinguish these two different type of interaction. Is this sufficient? So we asked ourselves, if we use now this model to screen novel molecules, can we correctly predict which could be antagonists and which could be agonists? So we apply this on let's say, commercially viable libraries of compound, and we find out 10 molecules that were very different from the known neuronal transmitter for adenosine receptor from a chemical point of view, but that our tool predicted to be antagonists. So what we ask ourselves is, from this, can we infer something if they are really antagonists? So I told you at the beginning that we need to look at the entire pathway that is activated once a GPCR is bound to an agonist or to an antagonist. And so we started to look into the pathway. And of course, there is a way to schematize this from a computational point of view. I mean, system biology has been doing this since several years. In other words, they have implemented the mathematical modeling of signaling cascade. So what we try to do is there a way that if we can infer structural information in the mathematical modeling of signaling cascade, and in particular, our structural information? So we build up a new tool that we call the structural system biology, that is a tool able to run system biology simulation, but that is able to implement also structural information via the calculation of the kinetic parameters that goes into the structural system, into the system biology approaches. So the pipeline is as follows. You can have a structural information coming either from simulation or from bitmap screening. You build up all the possible complex between the receptor and the ligand. You apply the discriminator to understand which equation to use, if inhibitory or not inhibitory. There is a prediction of kinetic parameter, always based on deep learning approach, and then there is the prediction of what come out at the level of the signaling. So we apply this on our 10 ligands, and what we find out is that there was in particular one whose predicted binding curve was similar to the standard. And indeed, we did the experiment. You see, above is our prediction and below are the experiment. And we were able to completely match the prediction. And we were able to find a new antagonist of adenosine receptor, completely different in chemical structure with respect to the ones that were being proposed below. And whose dose response curve, predicted dose response curve, was exactly the same of the one found into experiment. So this was for us a very important, let's say, proof of concept that it's possible to combine two key aspects of drug design, namely single molecule traditional drug design approach with system pharmacology, because from one side, the system molecule traditional drug design has a very high specificity and a very, let's say, detailed understanding of ligand target recognition, and also the possibility to include, for instance, the effect of structural modification, for instance, the ones induced by mutation. But from the other side, when you only apply this single molecule approach, you do not, you have a limited understanding of the impact of the ligand at the cellular level. In this case, at the subcellular level. So we managed to combine these two aspects, and this is how we think it is possible to go toward precision therapeutics, and what we are doing in collaboration with pharma company. And the idea is that when you build together all this aspect, of course, you can introduce not only the very detailed structural data, but you can introduce for instance, the characterization of genetic variation. And if you are able to introduce the impact of genetic variation, it means that you can introduce also individual variability. And this is what makes us believe that we can go, thanks to this multi-scale approach, toward personalized medicine. Of course, we have done this only on adenosine receptor, but I mean, this is, as I told you, as a proof of concept. Now, I just want to conclude this aspect by showing you this is a network about based on the topic connection between the publication in the fields of neuroscience in 1997. As you can see, it was a pretty much localized network. By introducing physics, biology, and computer science, the networks of topics in the publication related to neuroscience expand. And this just to show how also our multi-level calls also for multidisciplinary, and the different kind of expertise needs to be involved to reach this multidisciplinary level.
What Giulia, can you draw to a close?
Yes. I'm closing. So we use also this expertise not only for let's say for the context of brain diseases, but we have done also several contributions in the fields of COVID. And one of the current grant that we got from the Human Brain Project is exactly for looking at long COVID aspect using molecular level tools. And I would like to acknowledge all the people involved in this let's say research, as well as the EBRAINS on which our tools are all implemented. And thank you for your attention.
Okay, thank you very much, Giulia. Super. So now we come to our last speaker, Sebastian Billaudelle. So can you put your screen, your slides up? So Sebastian is a PhD student at the Cortical Institute for Physics at Heidelberg University. And he's working in the electronic vision group, which is working on neuromorphic computing, and was founded by Karl-Heinz Meyer and is now headed by Johannes Schemmel. And so he's going to be telling us something about basically the brain-inspired multi-scale simulations, and particularly training physical models of deep spiking neural networks.
Thank you, Rebecca, for the nice introduction. Maybe a quick note: I have been promoted to a postdoc position this year, but this, only a small side note. So in the following 10 minutes, I would like to walk you through the methods we use to train physical models of deep spiking neural networks, SNNs, on our neuromorphic system BrainScales 2. And that neuromorphic system, you can see a photograph of one of these neuromorphic chips on the left-hand side. And BrainScales 2 is a mixed signal neuromorphic system, and mixed signal means that we use both analog circuits and digital circuits as well, both wherever they are, they best apply. And BrainScales 2 emulates the dynamics of neurons and synapses in physical models, and it does so by applying a 1000-fold acceleration in contrast to biological real time, and you will see an example of this in a few seconds. The idea of physical emulation is that we can start from observing biological neurons, for example, their dynamics, and we can then try to find a model, for example, based on differential equations that describe those dynamics in detail. Here I show an excerpt of the Hodgkin-Huxley equations. Those are not implemented in our system, but only serve as an illustration here. And already Hodgkin and Huxley noticed that you can find an equivalent circuit diagram that behaves exactly the same like the differential equations you can formulate. And our approach is to use those equivalent electrical circuits and bake them into microelectronic systems. For example, the one you can see on the left-hand side. And each of these Neuromorphic BrainScales 2 chips then contains 512 neurons. Those neurons are analog circuits. You have voltages representing voltages and currents representing currents. They implement the leak integrate and fire, or even the adaptive exponential integrate and fire models. And you can see some recordings we have taken from those neurons in the bottom part. So we can configure them to a wide range of dynamics, for example, bursting, adaptation, and also transient spiking. Here you can also see an example of this accelerated timescale. While those patterns emerge in biological neurons on the timescale of hundreds of milliseconds, here they happen on the timescale of hundreds of microseconds. And this is something we would like to use for efficient computation. To form networks of these analog neurons, we also need synapses. Per chip, we have 512 times 256 synapses, and they basically form event-based in-memory compute arrays. And each of these synapses has a local weight memory with a 6-bit weight value, and you can also assign a sign to them. And one very important feature of these synapses is that they are plastic, and you can program a wide range of plasticity rules through on-chip plasticity processors. But a lot of this complexity we can just brush away for the moment and simply interpret the system as a collection of analog circuits that allow you to form networks of spiking neurons, and you can route event between them. And the question I want to answer now, or I want to talk about now, is the question on how do we train a physical model of deep spiking neural networks? And a lot of the challenges you stumble upon are already solved or have been solved in literature. For example, the spatial credit assignment problem is the question of how do we have to change a synaptic weight to have the loss function that describes the task we want to solve to minimize the loss function. And this is, for example, solved in artificial neural networks already by the backpropagation algorithm. If you have SNNs, you also have information propagating through time, adding a different dimension to that. And we also have to solve the temporal credit assignment problem here. And the idea, or one possible way to solve this, is to simply write down a two-dimensional compute graph, as it is shown here. Time flows from left to right, information propagates from bottom to top. The green layer emits spikes to the blue layer, triggering postsynaptic currents. Those are accumulated on the membrane, and the spikes originating from the blue layer then are forwarded to the red one. And you can simply walk back that compute graph to calculate the gradients, and this would be called backpropagation through time. The spiking nature itself also poses an issue. Gradients can only be estimated when we have a spike, and spikes also introduce discontinuities in the parameter space. And a lot of this can be solved by employing surrogate gradients. That means that we simply assume a different gradient for the thresholding operation, for example, as is shown here at the left side of a sigmoid. And with all of this now, you can train time discretized simulations of spiking neural networks, for example, through PyTorch. But with this physical system we have, the question arises, how can we calculate a gradient of something that we can measure, right? We can measure a voltage or spike trains from our system, but we cannot directly calculate a gradient for that. And the idea here is that we simply build a compute graph based on what we believe is happening in the system. We simply assume ideal dynamics, but this might not always be the case, because analog circuits might deviate from these ideal dynamics, for example, through variations in the manufacturing process. And we tackle this by continuously injecting measurements from the system into the compute graph, and those data are then, for example, the membrane potentials or the spike trains. And you can see some measurements here. time flows from bottom to top. This is a time window of only 340 microseconds. Of, I think, 256 neurons are shown here, and they participate in a recurrent network doing keyword spotting. And you can see that some of these neurons are inhibited. The membrane potential is basically depressed, indicated by this blue tone, and excited neurons are marked by brighter colors. And whenever those neurons reach their threshold, they emit a spike indicated by the red dots. And based on all of this, based on this compute graph, we now can calculate gradients for this physical system. And we do so by abstracting a lot of the intricate details in a high-level programming interface. This is only a preliminary version, but another one is already being deployed, I think, at the moment. You simply write down your network structure like you would do in any other network written in PyTorch. You can then inject data into this network in form of a PyTorch tensor, and you get out measurements. And this is all processed on the neuromorphic system. Then you can then write down a loss function. This might be an arbitrary, albeit a differentiable loss function, and you call the backward function on that to calculate the gradients. And we demonstrated this, for example, on the MNIST data set. Here we converted the pixel intensities into input spikes, as it is shown here. We then injected those into the neuromorphic system, where we recorded the hidden layer activity in form of spikes, and also the output neurons membrane traces. Those are 10 neurons, and the most active neuron indicated by a high membrane amplitude or potential. This would then be the classification answer of the system. And you can see that in all of these cases, the system correctly classifies the shown images. This network we implemented is quite small, only 246 hidden neurons, but yeah, the performance we could reach is basically equivalent, with only minute discrepancies, to a purely software-based simulation showing the reliability of the analog neuromorphic system. And if you have a closer look at these membrane potentials we recorded from the system, you can see that we don't have to wait for this whole period of, let's say, 35 microseconds until the answer can be read off, right? And this, basically allowed us to calculate the classification latency to less than 8 microseconds. And when you now basically insert an artificial reset of the membrane to the resting potential after those 8 microseconds, after you know the response of the network, you can then simply inject new images with a reduced spacing. And those the spacing resulted in less than 12 microseconds, which is equivalent to more than 84,000 images per second classified by this analog neuromorphic system. Considering the power consumption of less than 200 milliwatts of this of the chip, this corresponds to 2.4 microjoules per classified image, which is quite competitive, especially considering the fact that this is the general purpose system that wasn't optimized for this task. We can then also co-optimize on regularization terms. For example, we can penalize spiking activity, which allows us to classify the MNIST data with less than 20 spikes in the hidden layer, corresponding to more than 90% of the neurons being silent for each shown image. As I already mentioned, when you have such an analog system, you might find inhomogeneous dynamics due to variations in the manufacturing process. On BrainScales 2, we can calibrate a lot of this, as it is indicated by those purple histograms for example, the membrane at the synaptic time constants. But here we deliberately detuned those parameters. And we could demonstrate that even for standard deviations of 30%, we could train our system reliably. The MNIST data set was only one example. Here we used recurrent networks to perform classification of spoken digits of the spiking Heidelberg digit dataset, and again reached comparable performance to network simulating software of a similar size. Okay, to summarize, the framework I have presented is agnostic to the coding scheme you use. I have here only shown membrane potential based classification, but you can also do this for spikes in the output layer. It is independent of the network architecture and the loss function. It is robust to effects you may find on analog systems, and it's easy to use through a high-level user interface built on top of PyTorch. The efficiency that we achieved was mainly due to the analog computation and the sparse event-based communication of the BrainScales 2 system. and the acceleration factor allowed us to implement these low classification latencies and the high throughput. I would like to end on a less technical note. This project, I think, is a nice example for collaboration also with outside partners, in this case with Friedemann Zenke from FMI Basel, and this work was supported through an EBRAINS voucher. And yeah, as a final note, BrainScales 2 is available through the EBRAINS collaboratory, so if you have any questions, also feel free to contact us. I would like to thank you for your attention, and especially my colleague Dr. Benjamin Kramer, with whom I performed the work on this project, and Friedemann Zenke, of course, Johannes Schemmel, the group head, and the rest of the Electronic Visions group. Thank you.
Okay, thank you very much, Sebastian. So I think we've had three really interesting talks today, and we have a bit of time for questions. So we have some in the Q&A, and I will start to read. So the first one is for Jean-François. Do twins have the same folding patterns?
In fact, if you have a pair of twins, they look a bit more like each other than a random pair of people, but they are still very, very different. But surprisingly, from time to time, you can find a pair of twins very, very similar that stay in the same attractor. So it's my answer.
Okay. So there's one now for Giulia. Is it possible to expand the proposed approach to other neuronal receptors?
Yes, of course, it is possible in the measure that you have data availability for them. So for some neuronal receptor, you have a lot of experimental information that render machine learning type of model very solid. For others, you have less. So for the ones for which you have a lot of data and you can build up, let's say, efficient machine learning based models, you can of course extend. For others, you might need to find other strategies.
Okay, so then here's another question for Jean-François. Is there a part of your research which is looking at whether it is possible to predict changes on the geometry of the solid sheet so that it could be used to predict rehabilitation success? One example could be TBI or stroke patients and predicting their pattern towards recovery.
In fact, the patterns I was talking about probably do not change even with this kind of rehabilitation. We observe that even with amputation, you do not have change of pattern, but the size of the fall is changing, and whatever you do intensively is changing, in fact, the size of the falls.
Okay, good. So we have a question now for Sebastian. How many words and numeric characters are trained by using a spiking net? How is the activation pattern selected? And then I don't actually understand the rest of this question. Do the 5th and 0 number graph follow a similar pattern? Perhaps you can explain that.
Yeah, so we trained on the full MNIST data set, only downsampled to 16 by 16 images. So we had 60,000 images in the training set and the 10,000 of the test set. If I got your question correctly. And for the spoken digits, I think it's roughly 5,000 waveforms, but I'm not quite sure, to be honest. On that order, thousands of but yeah, you can check out the SHD dataset, the publication.
And in terms of selecting activation patterns?
Well, we used those. So for the activation functions, we relied on the leak integrate and fire dynamics implemented in the circuits. if again that was the question you were asking.
Okay, so now I think I'm really at the end of the questions. Okay, sorry, there is one more question for you, Sebastian. It's to ask how you would implement non-image data to train the model.
That question might have come up before I showed the spoken digit example. So we can basically train on any data you presented. You can write down any differential loss function in PyTorch that is based on the spikes generated by the system or the membrane potentials measured from the system. And then you can optimize that loss function.
Okay, good. Well, I think we can come to a close now. It's 5:30, so thank you everyone for attending this webinar. And if you want to see the replay, you can join the Human Brain Project YouTube channel. You can see the link here on the slide. And if you have any questions, you can also contact the Human Brain Project outreach at the email address given here. And the next webinar will take place on January the 24th, and it will be about exploring the mechanisms of consciousness disorders. So thank you once again to our 3 speakers, really great presentations, and thank you for your participation. And I wish you all a good day or evening.